Welcome to GATB 2020 MCQ practice series, where you can practice MCQ with me. And today's topic is cell biology. This is first cell biology practice series. In this series, we would learn about specific concepts of cell biology through question answer solving. Now, these concepts or these question answers actually came in JNU CEB. So based on previous year pattern and based on the important topics, I have created this question set for you. So this would really help in your last moment preparation, not only for GATB 2020 but for any other kind of MSc level entrance exam or even for CSI or NET. So let's begin the test. Question 1 says, cells in cell culture will absorb amino acid from the surrounding culture medium. If radioactively labeled glycine is provided in the culture medium, in which organelle will the radioactivity should be seen first? So obviously you have given some amount of radioactively labeled glycine as an amino acid in the cell culture media. Now this radio labeled glycine would be incorporated inside the cell, right? So glycine is an amino acid. Obviously, at that point of time, some amount of proteins are getting translated on the top of endoplasmic reticulum membrane, right? Some protein are already getting translated. So the mRNA is there and protein translation is taking place at that moment. So when protein translation is taking place, some amount of radioactively labeled glycine can get incorporated during this protein synthesis process, right? So you can understand that first of all, endoplasmic reticulum would be the organ where you would see the radioactivity. Now after that, from endoplasmic reticulum, it would be delivered into Golgi apparatus, right? So after that, Golgi apparatus would be the organelle where you would see the radioactivity, and if it's a secreted protein, it would follow the secretory pathway. But anyway, first endoplasmic reticulum and then Golgi would be the correct answer. So this question was based on the basic concept of vesicular transport that you need to learn about IIT JAM. Question 2 says, foot and mouth disease virus is the pathogen that causes foot and mouth disease in livestock. All analysis of the genetic material of O strain of FMDV showed that it contains 1996 adenine, 2131 guanine, then 1642 uracil, and 2365 cytosine base. Which of the following describes the genetic material of the virus? Single-stranded DNA, double-stranded DNA, double-stranded RNA, single-stranded RNA. First of all, looking at this question, you can clearly say these two options cannot be true. Both of these options are false because here they talk about uracil and here they talk about DNA. So either this option would be C or this option would be D. Now look at this. If it's a proper circular DNA, circular RNA, if it is a double-stranded, then what happens, guys? Then there would be enough amount of pairing happening, right? So every A would be paired with T, every A would be paired with uracil and G would be paired with C, right? So this pairing would be there. So equal amount of G and C, equal amount of A and U should be there. But look at A and uracil, right? Adenine and uracil are not equal in this particular given case. So the only option is it can be a single-stranded RNA, then only this can happen, right? So this question is based on simple idea about adenine, guanine as a nucleotide and the bonding between them. Then which of the following statement correctly describes the intermediate filaments? They are only cytoskeletal elements that are not composed of protein. Intermediate filaments are principal component of the cell cortex. The filaments are always found outside the cell. The filaments help the cell to withstand mechanical stress. Actually, we have to identify the correct statement. The correct statement is statement D. Now look at the statement A. they are only cytoskeletal element that are not composed of protein. This is false because intermediate filament is also composed of protein, just like actin and microtubule. Intermediate filament are principal component of the cell cortex. It is wrong because in the cortex region, in the periphery of the cell, you generally see actin based microfilaments. Okay, so this is false. Now these filaments are always found outside of the cell. No, these are not found outside of the cell. This is an internal structure, so this is a cytoskeleton, so it should be inside of the cell, so it is also incorrect. That is why fourth option is correct. So this particular question tests your understanding about intermediate filament, just the basic understanding, and also it is important to know that intermediate filament give us mechanical strength. So for example, if we stretch our epithelia, it does not tear off because of these intermediate filament. That's a very important concept. Question 4 says, which of the following cytoskeleton elements are associated with muscle contraction? Very important. We already know that muscle contraction requires actin and myosin based interaction, right? So myosin cross bridge formation is very important for muscle contraction, right? The sarcomere shortening. So obviously microfilament would be the answer, because this is a very simple question, but as we know, actin interacts with myosin, so we always want to find actin in this question, and we would sort of overlook microfilament from this question. From that angle, I have just generated this question. Now there is another question which says the table shows the number of cells at each stage of the cell cycle of root tip meristem. So there is a list given, and another word is given that one complete cell cycle takes 24 hours. Good. The number of cells at each stage is proportional to the time spent at that stage. So whatever time spent at that stage is proportional to the number of cells. What is the length of the time spent in metaphase? So you have to calculate that. So in metaphase, how much time you are spending? In metaphase, you are spending 12 minutes of time, right? 12 minutes of time. So this is important. So in metaphage, you are spending 12 minutes of time, and the fraction is 12 by 200, the fraction of time spent in the metaphase, and that would be multiplied with 24 hours. So it would be in some sort of minutes. So what you are going to do is like put minute, 60 minutes along with it. So if you do the calculation, so it would come something around, what should I say, 86.4 minutes. You can do the calculation by yourself. Then a student examined a cell in a garlic root tip and obtained the following data. So you can see the following data. Now, what is the percentage of cell that examined in which chromosomes are visible and consist of a pair of chromatid together? So a pair of chromatid which are held together is found in metaphase, right? So we have to talk about metaphase. So we have to calculate fraction of cells in the metaphase. So you can see roughly the total number is around 1000, and in metaphase it's like 18, right? So overall, you can understand the percentage would be something around 1.8%, right? So this is the correct answer. Now, which one of the following is not true about smooth endoplasmic reticulum, right? स्मूथ एंडोप्लाज्मिक रेटिकुलम के बारे में कौन सा वाला स्टेटमेंट ठीक नहीं है, तो वही हमको देखना है. स्मूथ एंडोप्लाज्मिक रेटिकुलम हेल्प्स इन ड्रग डिटॉक्सिफिकेशन, स्पेशली इन केस ऑफ लिवर. स्मूथ एंडोप्लाज्मिक रेटिकुलम have phytochrome P450 which is helping drug detoxification. ACR involves cholesterol metabolism. Yes, it is. If you want to learn about the importance of ACR, you can click on the video in the I button. ACR helps in phospholipid biosynthesis. It is also true because phosphatidic acid biosynthesis takes place in the smooth endoplasmic reticulum. Lastly, ACR helps in membrane protein folding. This is incorrect because RER help in membrane protein folding because RER have chaperones inside them, right? So this particular question requires you to understand the basic functionality of smooth endoplasmic reticulum as an organelle. Question number 8 suggests which of the following is not true about rough endoplasmic reticulum. Rough endoplasmic reticulum help in protein folding. It is true. Rough endoplasmic reticulum is a storage site for calcium. It is also true because it's like a reservoir for calcium, and it has several millimolar of calcium inside the cell, whereas in the cytosol the level is nanomolar. RER contains chaperone. That is also true. In the last question we have seen. And RER helps in Ross combat. It is not true because Ross combating mechanism is not present in RER. So this is the correct answer. So again, this kind of question requires your basic understanding about the cell organelles. Question number 9 suggests which of the following gas or gases function as signaling molecule in the human nervous system. This question was already asked in JAM and JNU twice, and it is very important. So the correct answer is nitric oxide or NO. Okay, because NO signaling is very important, and it is important for smooth muscle contraction, and it has been shown to be very crucial. Okay, so if you want to learn about more NO and cyclic GMP pathway, you can click on the link in the I button. Paracrine signaling is involved in which of the following processes? Chemical signals that can travel long distance between cells, autostimulation of T helper cells, hormonal communication, synaptic transmission between two excitatory neurons. So the last answer is true because this case, the two synapses, the cells are from the dendrites and the axon is from two different cells, right? And they are pretty much juxtaposed. But in case of hormonal communication, the cell that is secreting the hormone is secreting in the bloodstream, and it goes long way and get received by the responder cell. So it's a endocrine signaling, right? Also in the first option they said about endocrine signaling. So both of these options are not true, and they said autostimulation of T helper cell with the cytokine that it has secreted. So it's a sort of autocrine signaling, right? So autostimulation is autocrine here. So this is also not the correct answer. So the correct answer is D. Moving on, question number 11. Which of the following has an antagonistic action of adenylate cyclase? Cyclic adenosine monophosphate, active GTP alpha subunit for G protein, protein kinase A, phosphodiesterase. So let's analyze the question. Which of the following has an antagonistic action against adenylate cyclase? Means which oppose the action of adenylate cyclase. So what does adenylate cyclase do? So if it is a membrane, so adenylate cyclase is a membrane bound protein. So adenylate cyclase's job is to convert ATP into cyclic AMP, right? Now there are certain enzymes that converts cyclic AMP and break down cyclic AMP. These are phosphodiesterase. So diester bond is broken by these enzymes. So phosphodiester bond is broken by these enzymes. So this is just antagonizing the function of adenylate cyclase. That means generating the cyclic AMP. Adenylate cyclase generates cyclic AMP, phosphodiesterase breaks cyclic AMP. So it requires basic understanding of cell signaling. Question 12 says the target cell of lipid soluble hormones such as steroid hormone are able to respond to it because which of the following statement? So read these statements carefully. The hormone evokes a wave of second messenger signaling and further resulting in kinase cascade. They have membrane bound cell receptors. The hormone receptor complex stimulates the phosphorylation or dephosphorylation of subsequent proteins in the signaling pathway. Only target cell express appropriate cytosolic receptor which upon ligand binding translocate into the nucleus. And you can understand the correct answer would be option D. All of these other options would be false. If you want to learn more about steroid hormone signaling or how intracellular receptor works, you can click the link in the I button. Which of the following is not a second messenger? So cyclic AMP, IP3DAG, cyclic GMP and nitrous oxide. Here the correct option is D because nitric oxide is actually a ligand in nervous system or smooth muscle contraction. So it works via cyclic AMP. So this signaling is nitric oxide, cyclic GMP and protein kinase G. Protein kinase G signaling. So this axis is there, but this is the ligand. Okay, this is not the second messenger. Again, that required a basic understanding of cell signaling pathway. In a signal transduction system, proper switching on-off of a response is important. GPCR signaling can be switched off by the following ways, except receptor internalization mediated by beta-arrestin, downregulation of the GPCR mRNA, GPCR phosphorylation by GRKs, and GTP exchange in place of GDP in the G-alpha subunit. So you have to understand which of these following methods would not switch off the response, rather than it would have different consequences. So D is the correct answer, because if you replace GTP in place of GDP, so this would lead to activation of the signaling. So it would mimic a on state rather than an off state. So this is a wrong answer. Question 14 says mutation in G-alpha subunit that does not allow GTP hydrolysis by GTPase activity would lead to all of the following, except prolonged activation of adenylate cyclase. It is true, right? Constitutive active GPCR signaling, a dramatic increase in cyclic AMP in the cell, occasional activation of the adenylate cyclase. So let's try to understand the situation. Here we have the G protein coupled receptor signaling in the membrane. Now generally it is stimulated by its ligand, and there would be G protein having G alpha, beta and gamma subunits. Now upon ligand binding, this would be converted to G alpha GTP, right? In this situation it is activated. Now ultimately this would activate further adenylate cyclase. Now adenylate cyclase can generate lots and lots of cyclic AMP, which is the second messenger in this pathway, and cyclic AMP ultimately works via pKa. You guys know already this thing, right? But look at this thing. So prolonged activation of adenylate cyclase. The question is, if this is on forever, what would be the consequence? So obviously adenylate cyclase would be hyperactivated, it would be on for prolonged duration. So that is why a lot of cyclic AMP would be also generated. So concentration of cyclic AMP would also increase. Now dramatic increase in cyclic AMP, that's why it's also a true phenomenon. Now constitutive active of the GPCR signaling, obviously overall GPCR signaling would be on for a long time. Now last option, the occasionally activation of GPCR signaling or adenylate cyclase, is contradicting to the prolonged activation of adenylate cyclase. So this is the proper answer. This is the wrong statement and this is the proper action, okay? Proper answer. When the cell releases a signal molecule into the environment and the number of cells in the immediate vicinity respond to that, the signaling type is paracrine signaling. So this kind of question is very important. Paracrine, endocrine, exocrine, which kind of signaling is a paracrine signaling? So they would give you an example and you have to identify this is what type of signaling, and they would maybe define the type of signaling and you have to understand the name, what it is. So this question is just for that. Question 16 says there is 14 units of DNA in the cell during G2 phase. How many units of DNA would be there in the G1 and S phase? So let's draw the cell cycle. Right now what we see in the cell cycle, we have G1. So forgive me for the proportion here, just imagine there, okay? So G1, G2 and M. So they see that in G2 phase there are actually 40 units of DNA. So obviously in the S phase there is a duplication, right? So obviously you can understand S phase it happened to be 40. So the 40 actually came from 20 plus 20, right? So in the G1 phase there would be 20 units of DNA which would be replicated and be twice in the S phase, which persists in the G2 phase as well. So obviously your answer is, so you have to identify how many units of DNA are present in a cell in G1 and S phase. So in G1 it would be 20 and in the S it would be 40. So the correct answer is C. Again, it tests the basic understanding of the cell cycle and the key events of the cell cycle. Which of the following description of mitosis is or are correct? DNA synthesis occur at the beginning of the prophase. The nuclear envelope breaks down in prophase and reforms in the telophase. The division of the centromere occurs in the anaphase. So you can clearly understand the option would be option B, right? Read this statement. If you don't understand, look at my cell cycle playlist in the description, okay? So you would find it in the I button. Click on that. Question 18 says which of the following is not an example of proto-oncogene? So proto-oncogenes generally help in growth. So we have to identify which of these following help in growth, and the molecule which won't promote growth would not be a proto-oncogene. For example, CMYC, BCL, XL, all of these help in cell survival and growth, right? So these are proto-oncogenes. Cyclin D1 obviously help in cell division, right? Cell division and G1 to S transition of the cell cycle pathway. So obviously it helps and promotes cell division. Lastly, BAX. BAX helps in apoptosis. So it's kind of a negative regulator of growth or negative regulator of division or cell survival. So BAX is the proper answer. So this is not an example of proto-oncogene. It's a tumor suppressor category. Okay, cancer can be caused by gain of function mutation in the proto-oncogene, loss of function mutation in the tumor suppressor gene, viral oncogene, all of the above. So this is a very important question because this tells you for what reason cancer can be caused. Most of the cases you might know option A and B are correct, but C is also correct. So viral oncogene can also cause cancer. So proper answer would be D. All of these are correct. If you want to understand what is proto-oncogene, what is tumor suppressor gene, want to have a very good concept of it, click on the link in the I button. Okay. In the case of irreversible DNA damage in the G1 stage, the tumor suppressor protein P53 can block cell cycle progression from G1 to S by inhibiting what are the things? So obviously they block G1 cycling, but the way they block G1 cycling is by activation of P21. So P21 directly block the cyclin D, okay, and P53 mediates this process. So both option A and B are correct. So C is the correct answer. Question 21 says, if a researcher add microtubule blocker and another drug for actin myosin cross-bridge formation blocker, then what would be the effect? The drugs are provided separately in two separate cell culture dish. So none of these experiments are overlapping with each other. So which stage of the cell cycle would be affected? So this is another interesting question because the stages that would be affected would be metaphase and cytokinesis, because in metaphase you need to separate the chromosome, and this is with the help of microtubules, right? They pull each other in the opposite pole, and that is why separation takes place in the anaphase. So if you put microtubule growth inhibitor or microtubule blocker, so what would happen? The microtubule would be either hyperstabilized or totally destabilized, so this would be a problem. And in case of cytokinesis, so you need to form an actin myosin ring, right? And that ring contracts and that leads to two different cells, right? And if you put a blocker such that the actin and myosin cannot interact with each other, then what would be hampered? The cytokinesis process would be hampered. Again, this question requires a very fundamental understanding of cell biology, so it's not so difficult. Question 22 says, cyclin dependent kinase phosphorylates dashed residue of the target protein. This particular question is asked in many exams many times. So let me remind you again, cyclin dependent kinases are actually serine threonine kinases. They are kinases in nature, they phosphorylate protein, and they phosphorylate protein in the serine threonine residues because they are serine threonine kinases. What is the name given to the structure which penetrate the wall of adjacent plant cell? It is very fundamental. Everybody know, we are studying from the class 10th. It is plasmodesmata, right? Now question 24 says, all of the following statements are false except IP3 is a second messenger but calcium is tertiary signaling molecule, right? So what is a tertiary signaling molecule? Nobody knows. G protein is always trimeric in terms of structure. No, it can also be monomeric. For example, RAS, right? We have talked about it in MAP kinase pathway. G protein coupled receptor gets activated when GTP is exchanged in place of GDP. So this is a correct statement. And the last one, gaseous substances can also work as a signaling ligand. So this is also another correct statement. So this is basically a MCQ type question. So both these options are correct. Now, nuclear membrane breakdown takes place at which phase of the cell cycle? So it is actually prophase. And in prophase what happens? Lamins, which are intermediate filaments, they break down. As a result, what happens? The framework on which nuclear envelope is there, it is breaking down. So it would ultimately lead to breakdown of nuclear envelope. So this is the overall question set. So I hope you enjoyed this video. If you like this video, give it a quick thumbs up. Don't forget to like, share, and subscribe. Thank you.